So I I choose this topic actually, drug discovery, because of the recent the ongoing pandemic situation. Because everybody is running after a drug, okay, and currently there is no drug. Couple of drugs are in the pipeline, and couple of vaccines are available, and there are a lot of doubts on all of those vaccines, mainly because of the side effect. And now the question is always, I mean, the general people are asking, it's almost a year, I mean, it's more than a year actually. So why don't we have the drug? It's, for a common man, maybe 1 year is a long time. And they think that we can have a drug in, in 1 year. But that's not the case. Actually, the drug discovery is a very lengthy process. Okay, it takes a very long time. Okay, so generally it takes 12 to 15 years to develop a drug. Sorry. Can you see my second slide? The long road to a new drug. Hello.
We can see your title slide only, sir.
Not the second slide.
No,
Right.
not yet. Now it's visible.
Now it is there.
Okay, so then that is not good, because that means in full screen you cannot see it. Anyway, so now it takes almost that long period, actually 12 to 15 years, and it cost more than $1 billion. So it starts with the project team, okay, and plan. So first you have to identify the disease you want to develop the drug, then you okay, that which protein I should target so that the disease can be recovered. Okay. So now, so this process actually it has a lot of I mean, it takes a lot of time. And as a computational scientist, your job is to reduce the time. Can we reduce the time frame from 12 to 15 years to say 5 to 7 years? That's a great achievement. If you can reduce the time frame by half, then you are reducing the cost. So then, of course, the cost of the product, that is the medicine, will also be less. So why it is so lonely? Because first we started with this basically early exploratory development first. Okay, so the team management sit together, they decide which disease they need to work on, and then what is the target, and then they try to find out what are the lead compounds, and then you synthesize those compounds, and then first you do the animal model study. Okay, so those are the early safety study. So mouse model you test it and all those things. First you test it, and then once you qualify, pass this early safety study, then you have to go for the human study. Okay, so you create a large amount of candidate medicine synthesized, and then you study in healthy volunteers. So that is the phase 1 trial. So if you pass in phase 1, then you go to the next level. That would be the phase 2. Okay. So there you have to use it on 100 to 300 patients. So now in phase 1 you can fail. You can fail in early safety study also. Basically in each and every step you can fail. Now if you fail in say early safety study, that is an animal model, then again you have to come back to the stage 1. Or if you fail in stage 1, okay, clinical study phase 1, then if you fail there, then again you have to come back to the first stage and then perform the whole thing. So if you pass phase 1, then you go to the second stage of clinical trial, and then after that, if you pass there successfully, then you have to go for the phase 3 trial. Okay, in phase 3, basically, you have to test it on 3,000 to 10,000 patients. Okay, and those are all random, basically selected. And then you generate all the data and you study these things, side effects and everything. You have to study it, the effectiveness, efficacy, all those things you study. And based on your result, then you should apply for the registration process to FDA. So this whole process actually takes almost 10 to 12 years. And you can fail in any stage. The problem is that 54% of the drug till 2004 actually. So 54% of the drugs, they fail at either clinical phase 2 or 3. So you see that if your drug fails at clinical trial 3, you already have spent 8 to 10 years. And then study fails. And again you have to come back to this project team and plan, this exploratory development stage. Think about it. So that is the challenge in drug discovery. It is not that easy. It takes a lot of time. So now as a computational scientist, we want to minimize this time period. And we want to give the lead molecule to the experimentalist that, okay, these are the magic molecules you should try. Maybe they may work on it. Because in computer, you can study this toxicological thing and other properties of the drug. And if it satisfies, then you say that, okay, so I find it to be quite interesting because it satisfies all the properties that a drug molecule should have. Okay, so then that will reduce the time frame. Because if an experimentalist, they have to basically, first the medicinal chemist, they have to synthesize thousands of candidates and then test it, screen it, and it will take a lot of time because synthesizing thousands of molecules is not that easy. If it is a new molecule, if it is available in the market, then it is fine. You can just buy it and use it. But if you have to synthesize, then it's not that easy. Okay, so that is the difficulty, and then our aim is to reduce that time frame. Now, how can we do it? Okay, so that is basically we can do it using various computational techniques for this drug design. So right now we have a database. Okay, so it has, these are the different components that's computed drug design process. So you have database. Database is nothing but small molecule databases that contains the 3D structure of these small molecules. Small molecules are the ligand molecules. That's the drug candidate. Okay, you can further modify it to be a drug. Okay, so basic building block is available. And then you also need to know the homology modeling and molecular modeling techniques, because often you may know the structure of the target protein, okay, but you may not also know the structure. You may have identified, okay, this is the sequence, but you have to predict the structure of the protein. Because experimentally determining structure is not that easy, especially using X-ray crystallographic technique. It's pretty difficult actually. And if it is a membrane protein, then the difficulty level increases further. So often you may face a problem that you don't know the crystal structure of the target protein. And in that case, you have to use various computational technique to predict the structure of the protein. Okay, and then you target that protein to develop your drug. Now, another thing, as you have now say billions of compound, okay, and you have to screen against that particular protein, and you have to find maybe 10 best candidate that binds very strongly with the protein. Okay. So from billions of compound to 10 compound, how do you do it? That is the challenge. In computer, you have to, because currently you have say 1.3 billion compounds actually there, the database. So you have to screen it, and you have to do it within a reasonable time frame. Okay, so we have to use various machine learning and deep learning techniques so that we can screen all these molecules against a target protein within a reasonable time frame. And this screening can be different ways you can do it: structure-based, shape-based, pharmacophore-based. So all these are different ways that you can do it. Now, another aspect that is very difficult is the target identification. Okay. So target identification, that means that which protein is you should target. I mean, which protein is responsible for this disease. Okay, the disease causes protein, and that you need to stop its function so that the disease is arrested. So that is another thing. And then once you identify the protein, then you have to identify the binding pocket where the drug molecule will bind. You have to find that thing also. Okay, so those are the things. Then you find out the number of, 10 based molecules or 5 based molecules, and then you should do this lead optimization. Okay, based on biophysical experimental data, you can modify the chemical structure, you can add something or delete something from it, some functional groups, so that it binds more strongly with the protein and also its bioactivity increases. So that way you can design it. Finally, once that is done, then you will go for, then you give this molecule to the experimentalist, basically a medicinal chemist, and they will go for the animal study. Okay, now, is it possible for an experimentalist to synthesize all these 1.3 billion compounds and do the biological screening in experiment? Yes, they can do it, but you don't know how long it will take. It will take a couple of years. So if the screening takes a couple of years, then you have to do this animal study, then the clinical study. So you can't get a drug within a reasonable time frame. Okay, so that's why we want to use various computational techniques to reduce this time frame, at least the initial screening phase. That drastically we can reduce the time. So there are two different ways. Basically, this computer-aided drug design can be done. One is the structure-based drug design. Another one is the de novo design. Okay, so when you know the crystal structure of the target protein, then you can use the structure-based drug design. Okay, and if you don't know the crystal structure of the protein, then you have to do the ligand-based drug design. Okay, and so it's generally today we are going to talk about structure-based drug design, because, and I will give you some example in the second lecture. Actually, I will give you an example from SARS-CoV-2, that is the current pandemic, what we have done and what other people have done, the drug thing. So the structure-based drug design, we started with first we have to identify the target and select it, and then we have to isolate it and purify the structure. Okay, and then you determine the crystal structure using X-ray crystallography. Okay, and then using computational techniques, you can visualize the protein structure and identify the binding site. Okay, and once you identify the binding site, then you can dock the small molecule into the binding site. Okay, and then you perform this experimental study, biochemical assays, and further testing. Based on those data, you can modify your ligand further. Basically, you optimize the structure so that it binds more strongly. Okay, so you perform all this lead optimization to improve the potency. Once that is done, then you can study the cytotoxicity. Okay, whether it has any side effect. So that you have to study, and the pharmacokinetic studies also you have to do that thing. Once it passes, then you have a drug candidate. With that drug candidate, now you go for the clinical trial. So these are the steps for structure-based drug design. Okay, so As a computer scientist, your job ends up to this cytotoxicity test. You can do that thing in computer also using various machine learning based techniques that you can do. Basically, it's a classifier problem. Okay, so you can do that thing. And next is this ligand-based drug design. Ligand-based, basically that relies on the knowledge of known molecule that bind to the biological target. Okay, so basically you know the structure and bioactivity. That is the IC50 values that you know. And these molecules may be used to derive a pharmacophore model that defines the minimum necessary structural characteristic a molecule must possess in order to bind to the target. And we perform this virtual screening based on the pharmacophore models, including drug property filtering. Okay. For that one, we have a zinc database that you can use. This is a free database that is available. You can. This is not commercial database. You can just download it and you can use it. And alternatively, there is another approach that is quantitative structure activity relationship or QSAR based model. You can use it that one also for further processing. Okay. So that means that is a regression problem. Okay. So in drug discovery, actually, you have a lot of applications of machine learning and deep learning method process actually. So regularly you can use those method to discover a new drug. And currently, the thrust is on how you can use these AI-based models to minimize the time frame and get a better drug candidate. And you'll be amazed to know that this Google, they have created one thing, this deep learning based method they have, deep neural network based model they have created. It's called DeepMind. Okay. So that they use this model to predict the crystal structure, not crystal structure, okay, they want to predict the three-dimensional structure of a protein from sequence. Okay, so now their efficiency is very high and accuracy is much too far we have done. So there is a quantum jump basically in its accuracy. So that's is kind of so last year basically they submitted this thing and it is published in Nature articles. So previously there is a competition called CASP. Okay, so what happens here? The experimentalists give you a sequence and you have to predict the three-dimensional structure of the protein from that sequence. And the experimentalists, in the meantime, they resolve the crystal structure. So they have everything. So you predict it and then try to match with the experimental result. Okay. And this gives us the opportunity for computational scientists to improve the method as well as the underlying physics to predict this three-dimensional structure of a protein from the sequence. Now, all those methods that previously been used is surpassed by this Google lab. Okay, so they use this deep neural network model, and the accuracy level is almost twice that what has been done in 2019. So there is a sudden jump. So that is the level of applications that people are doing of AI in drug discovery. So it has a lot of implication in drug discovery because, as I told you, that in many cases that you have identified the disease, but you don't know the crystal structure of the protein. But it is easier to get the sequence of that protein. So in that case, you can use that method and predict the crystal structure of the protein, and then you can do the molecular docking study and go for drug discovery. That makes life very easy, actually. And that code is available free of cost. You can search in Google and you will get it. So you can use that method also. So I'm not going to do much of this, but I'm going to talk about the virtual screening. So now I told you that for drug discovery, for say structure-based drug discovery, you know, for example, you know the crystal structure of the protein, okay? Or maybe you have predicted the structure of the protein. And you have identified the binding site of the protein also. Okay, you know where exactly you want to fit the molecule in the protein. So now the question is, uh, you don't know which one is the best molecule. You have to identify it. And you will start with a bigger or larger database. Okay, so the current database this has 1.3 billion compound, and you want to screen that thing. So you can imagine how long it will take time. Okay, so how do you do that thing? So there are various ways you can do it. All the method has accuracy level, and generally modeling means, okay, modeling means that there is always a compromise between speed and accuracy. You cannot make a method which is the most accurate at the same time it is the fastest. No, it's not possible actually. Molecular modeling is basically approximating and try to make a fine balance between the speed and accuracy. Okay, so now we have this method called virtual screening. So this is a in silico analog of biological screening that experimentalists do it using SPR method. Okay, that is scanning probe microscope. So they use it for this kind of study. Now here in virtual screening, your aim is to score, rank, or filter a set of structure using one or more computational procedure. Okay, so we use it to help decide which compound to screen experimentally. You don't want to screen all these 1.3 billion compound experimentally. No. Which libraries to synthesize? Okay, if you pinpoint them that, okay, these are the 10 molecules, I find it to be very interesting and could be effective. So they will just only synthesize those 10 molecules. Or if it is available in the market, then you can buy it from the market and go for experimental study. Okay, and then which compound to purchase from an experimental company, and then to analyze the result of an experiment such as high throughput screening run. Okay, so so you use it for those kind of purposes for the virtual screening. So here, this is a very fast method. At the same time, you should remember that if it is a very fast method, then its accuracy level is also very less. Okay, but that's why this method is not used to identify a single molecule. No, we just screen it and rank all the molecules. We can rank all these molecules based on their binding free energy, basically how strong they are binding to the protein. We just rank them and then we make a cutoff. Okay, above this binding free energy, we should consider for experimental study, or we should study further for toxicological study. Okay, so you can do that way actually. Its job is like that. It does not exactly tell you among these 10 which one is the strongest. No, you cannot tell it with certainty. For that one, we have to use better method. Better method means longer time. So that's the thing. Now we are going to talk about basically the molecular docking part. Okay, molecular docking is basically is very important in the case of molecular recognitions. Basically, how the drug protein recognize each other. Generally, it consists of different non-covalent interactions using such as hydrogen bonding, electrostatic interactions, and hydrophobic interactions. So those are hydrophobic interaction means it's entropic in nature. So that also plays a critical role. So those are the different forces that controls these protein drug interactions, and we want to understand that which force is dominant for this binding. Can you optimize it further? Okay, if one force is less, can you optimize it? Okay, so those are the different kind of thing we want to study from computational, various computational technique and optimize the drug further. Now, for the drug discovery, first thing we need to understand, we have to see the drug molecule from the perspective of a protein, okay, from the perspective of a biology. So as a chemist, what do you see? You see, say, this is aspirin. Aspirin, these are two common drugs, actually. Aspirin is very common. And the right-hand one is the sildenafil, which is the commercial name is Viagra. So as a chemist, you see it as a bunch of atoms, and they are connected by single bond, double bond, okay, and ring structure. You basically see it like that. And now, so one thing is that if the structure is presented like this way, okay, single bond, double bond, like all, this way, okay, this H3C ring structure, then you cannot use it for machine learning. You can use it, no? So we have to convert it to a certain way so that I can use natural language processing technique, NLTK, we can use it, okay? So for that one, we convert this thing into this Miles format, simplified molecular line entry system. Now you see that if it is like that, then you can use NLTK, natural language processing techniques. You can use it. So this is how a chemist can see it, okay? And then as a computational scientist, you can represent it like that so that you can use it for machine learning study. But when you want to develop a drug, you need to understand how the protein sees this chemical compound, okay, this foreign compound. So does not see it as a single bond, double bond, all those things. No, it sees this is like a surface, okay, as a van der Waals surface, and on it you have basically the charges or the potential is mapped onto this surface. So you have the red are negative and the blue are positive. So it will see like that. Now you can understand one thing looking at this. Okay, aspirin, you see that there are a lot of negative charges on the surface. So now, if you want to target, if you want to fit this molecule to a against a protein, then what to do? Of course, you will try to find the binding site which is actually more positive, because this one is negative, and if that surface is positive, then there could be a stronger binding. Okay, so those kind of intuition you have to use it while you want to design a drug. Okay, and in the below picture, we have shown the how does the binding site in the protein that looks like, okay, in terms of hydrophilic or hydrophobic. So green are hydrophilic, that means those are water loving, those are exposed to the water because in your body most of it is water or membrane. No? So if it is a cytosol, that means it's water environment. So most of these amino acids are exposed to water. So they love to interact with water. And certain amino acids, they don't want to interact with water. They are hydrophobic in nature. So they are from the core of the protein. If it is exposed to the water, the protein will unfold. Okay. So that is also hydrophobicity. That's why it is entropic in nature. So this is how it looks like, and you have to fit it to this binding pocket. So then this molecular recognition that I told you, basically that helps you to understand this dark discovery process, and you can block the recognition, that is inhibitors, antagonists, and or emulate the recognition, that is agonist, recombinant protein, those are the things. So overall, this picture actually tells you, as a layman perspective, what is molecular docking. So docking attempts to find the best matching between two molecules so that they are snugly fitted. And here, if you see that this is the left hand, if you look at this figure, so the protein is there and the binding site is shown as a red circle, and the ligand, that's the dark molecule, is shown as a green, okay? And your job is to just insert this ligand into this binding pocket, this cavity that you can see it, you have to place it. Now, when you place inside this cavity, it can have different orientation, and different orientation means different energy. And you have to find out the orientation for which the energy is lowest, that is most negative. Your job is to maximize the interaction and thereby minimize the total energy of the system. Because lower the energy, more stable the system is. Okay, so lower potential energy corresponds to more robust and stable structure. So that's your job. Okay, so now it can have so many orientation, and you have to calculate for each orientation the interaction energy on the fly and store it and find out for which orientation it is the lowest energy. Okay, so that is basically the molecular docking. And this docking process, we are doing it in computer, but it starts within a second. But in computer, it takes so lot of time. So that's the job. And aim of molecular docking is to achieve an optimized conformation and relative orientation. That's what I said. Uh, Between the protein and ligand, such that the delta G, that is the binding energy or the Gibbs energy, is minimized. Okay, and it can tell you whether the two molecules interact with each other. If the delta G is positive, then it means that the drug does not bind with the protein. So that's a wrong. That means that's a wrong candidate you have chosen. Okay, and if it binds, then your job is to find the orientation that maximizes the interaction and which minimize the total energy of the systems. Okay, so that's your things. So an overall goal, given a protein structure, predict its ligand binding. That is the goal of the molecular docking study. So now we will go into this. Talking generally, there are different models that so actually old problem. It started in 1890 by Emil Fischer, and the model is called lock and key. Because you see that you can open a lock with a single key. Multiple keys cannot open a single lock. Okay, that particular lock can be opened by a particular key only. So this is the same thing. You have a protein that is your lock and the ligand is your key. Only that particular ligand can bind to that protein. Okay. And then came the induced fit model by Danny and Postland. It means that the protein can adopt some structural changes. And then in 2003, the better method came, that is conformational ensemble method. We'll talk about all these various methods. So I told you about this key and lock theory. So basically the bluish one is your ligand, and this yellowish one is the protein. So it's just snugly fitted. Okay, so there is no structural changes or anything. So that is your key and lock method. But in induced fit, you see that the white one, first look at the left-hand panel picture. Okay, so the white one is your ligand and the blue one is your protein. Now you see that, of course, the ligand cannot fit it with the protein snugly. Here, in this case, the ligand also changes its shape, that is its three-dimensional structure, and the protein also changes its three-dimensional structure. Now, both basically changes their structure such a way that they can bind very strongly. Okay, so there is some small conformational change until an optimal fit is achieved. This is called induced fit theory. Now, in key and lock theory, there is no such thing. So it means that here both the ligand and the protein both are rigid molecules. So the rigid molecule problem is very easier to solve because your number of degrees of freedom reduces drastically. But when you let everything is flexible, then the number of degrees of freedom increases, and that makes your problem more complex. Okay, so there's a various complexity arises in drug discovery that you need to handle using various computational techniques. And then you have the conformational ensemble method. So here you have a bunch of protein structure, the same protein, but each structure has different energy, slightly different energy. So it has this kind of energy, this underlying energy structure looks like this. Now, if you look at this figure, you can see the problem here. The problem here is that there are multiple minimas, and each minima is separated by a high energy barrier. So now your job is to go to the global minima. Search the global minima. So it's an optimization problem. But how do you do it? If I want to study the time evolution of this drug molecule movement, that I want to study how it is moving to the global minimum. So you can imagine how difficult it is because the energetic barrier is so high, it may not be able to overcome that. And your simulation may get stuck there for a long time before it can overcome that barrier. Okay, at room temperature, your energy is 0.56 kcal per mole. And if the barrier height is more than that, then how can you overcome it? Okay, so those are the problems. But if I want to address this problem in such a way that, okay, I will take the various structure of the protein, the same protein and various structure that corresponds to various minimum. And then I dock the molecule in this various protein structure. And then I try to find out for which case the binding affinity is the lowest. Lowest means more negative. Okay, it's not in absolute term. It's more negative each one. So that would be the best protein ligand structure. Okay, so this can actually reduces a lot of computational resources because you basically the structures are previous. I mean, you just do a short simulation and get these underlying different conformations that corresponds to different energy, and then you are talking it. It is better than, of course, than the key and law principle because there everything is rigid. So that's the thing, and with the rigidity, I mean, how the complexity actually increases, as I said, that if it is a rigid molecule, then actually it's very, I mean, the number of degrees of freedom actually is less, so it's a less complex problem. But when everything you make flexible, then the number of degrees of freedom increases, and that makes basically very difficult to study. Okay. So now this is the problem. Okay, various problem. Basically, a bottleneck in drug discovery is the 3D structure of complexes are rarely available. So that means you need to predict the 3-dimensional structure. But recently, this problem has been addressed to some extent by this DeepMind software. I mean, this one. You can do that thing. And next is that is one thing. And another bottleneck is the flexibility. In principle, proteins are plasticity. They are very flexible, and drug molecule is small, so they are also very flexible. Both are flexible. So how do you dock? So if they have both are flexible, then the number of degrees of freedom increases rapidly. So none of the docking methods are actually the best method. Okay, so we can use a small flexible based method, and then you can run molecular dynamics simulation method where everything is flexible. So you can use two stages of studies. So the first stage I'm talking about molecular, I mean this docking, and the second lecture will be on the molecular dynamics, the large scale simulations part. So now this docking, how can you do this? Various ways you can do it. One is the shape complementarity, okay? That way you can do it. And another one is the chemical complementarity. So shape complementarity is the primary criteria for evaluating the fit in the computational docking of two candidates. And chemical is and physiochemical complementarity are also important criteria because see that you want to place your molecule near to the protein molecule such a way that your drug molecule can form a hydrogen bond with the protein, or they can have aromatic interactions or hydrophobic interactions. So you have to place your molecule such a way. So these are the different physiochemical things that you look into. So that is one way of doing it. And the other way that you should always remember here is that the energy, okay, energy dictates the molecular association. You have to make sure that when the protein and the drug molecule form a complex, the total energy should be more negative. Okay, more the negative, more the stable the structure is. Okay, so the goal of the computational docking is to find the 3D configuration of the complex that minimize the energy. So if you look at this right hand panel picture, so of course you see that I have highlighted with the greenish color. So that one is that shows the lowest energy. So that would be the best protein ligand docked structure, protein docked structure. Okay, so that you should remember the thing, but that could also be misleading. We'll talk about it. So, so ligand docking is fast heuristic approach with two key components. Generally, we do it. First one is the scoring function. So when you are placing the protein, I mean the ligand, the drug molecule in the binding site, then you want to calculate the binding energy. Okay, because that is your criterion. You are rotating it, various position. You are evaluating the energy, and you are trying to calculate the which one is the lowest energy. So this evaluation of energy is done by the scoring function. So that very roughly approximate the binding affinity of a ligand to a protein given a binding pose. Okay, pose means the orientation of the drug molecule in the binding pocket. And next, what you need to, what other component is the search algorithm. As I told you that you have to, because inside the binding cavity, the ligand molecule can adopt various orientation. Okay, and various orientation means various energy. So you have to have a search algorithm that searches for the best scoring binding pose for a given ligand. So it should be exhaustive. Okay, so each all possible orientations you have to start and evaluate the energy. So that is the search algorithm. So you can use genetic algorithm or whatever different ways that you can use it for this search algorithm. Okay. So now I told you that the main problem with the protein ligand docking is the flexibility, because proteins are flexible, ligands are. So generally, and I also tell you that modeling means approximation. You want to approximate so that you make a fine balance between speed and accuracy. So here you approximate the protein is rigid and the ligand is flexible, and you know the approximate binding site. Okay, it's not blind docking you are doing that. You can anything. Okay, but in reality, ligand mobility, protein mobility, and then water molecules all play a major role in determining the binding affinity. So that's why molecular docking is good. It is a first step. It is not the final step. Okay, we have to supplement molecular study by large-scale MD simulation study. Okay, so that is the thing, and so generally, how we treat this molecular flexibility in ligand docking. So docking is basically three different categories. So one is the rigid body docking, where both the protein and the ligands are rigid. Then the semi-flexible docking, so where only the ligand is flexible and the receptor, that is the protein, is rigid. And then flexible model, where both the protein and ligand are treated as flexible, which is the most accurate method. The flexible method is the most accurate method, but it is most time-consuming also. You have to use your method based on the availability of computational facility. Okay, so generally, this is the thing. Initially, receptor and ligand both are rigid. Then there is the fast and simple. On the other hand, the advanced approach, that is the receptor and ligand flexible, that is the slow and complex method. Okay, this is how it increases. Generally, most of the currently used docking softwares, they use the in between method, where the receptor is, that is the protein, is rigid and the ligand is flexible. It is easier to handle the flexibility of ligand because drug molecule have generally 100 to 200 atoms, but protein has couple of thousand atoms. So if it has couple of thousand atoms, so you can imagine the degrees of freedom will be increases 6n. Okay, because there are 3 rotational degrees of freedom and there will be another 3 translation degrees of freedom. So there will be these 6 n types of 6 into n degrees of freedom. So it increases significantly. So that's why we want to make protein as rigid and the ligand as flexible. So that way you do it. Then you run large-scale MD simulation where everything is flexible. So we want to do it in this two-step process, basically, for the drug discovery. So now this is how I already tell you that how it is difficult actually. When it's flexible docking, then it's basically an intractable problem. And for rigid docking, it is much easier. Okay, so everything is you can track it. So we use various ways to for handling ligand flexibility, because I told you that generally we use ligand as flexible and protein as rigid. Most of the softwares, those are available. So how they do it? So ligand flexibility, you can take it by taking ligand ensemble docking method, fragmentation method, and stochastic conformational search method. In ligand docking method, the same ligand, okay, you have created an ensemble of conformations. How do you do it? You can rotate the angle, okay, the phi angle of the this this ligand molecule. So then you will have multiple conformations. So then, so you created say 1,000 such conformations by rotating the phi angle. Okay, and then you dock each of these conformations to the protein, and you evaluate the energy. Then you find out for which for which orientation that you have basically rotated the molecule. No, so for which case the energy is lowest. So then you know that okay, that is the best possible orientation of the ligand molecule. So that way you are taking into the flexibility of the ligand molecule. And fragmentation is basically you don't the drug molecule entirely you don't block it, rather you fragment it into different components. Okay, and then you just make a small fragment, you place it one fragment, evaluate the energy, then you add the second fragment, then you add the third fragment. That way you can do it. Okay, so that way also basically you are introducing some kind of flexibility in your system. Okay, so that's what I talk you. Each subpart basically you is docked independently. You have separated and then you have assembly, basically reconnecting all the subpart using the chemical bonding, and then you so that it got a geometrically correct conformations. Okay, basically you need to know whether there should be a double bond or single bond or triple bond, that you need to do it. Okay, so that method we use, or we can use the incremental based method. Okay, so we start with an initial core docked in the active site, and new fragments are progressively added, and we minimize the system. Okay, so that treatment is terminated when the entire molecule is formed. So that is one thing. But incremental method, the problem is, say suppose there are three different ways. I have shown you here one example. Okay. So this fragment that you are starting, these three different positions you are starting, and you can see that you just start with that position A, okay, and incrementally you are adding it. Then you reach to a final conformations where you have added all the fragments and it nicely fitted. So that is fine. But if you say take the position B and then you are adding it, then again it fitted. But which one is better, A or B? In which case it is fitted better? I mean, in which case it forms large number of interactions. You can see that, of course, in case of A. Now, if you start from position C and eventually you are adding all the molecules, then you can see that after two steps, you cannot add another fragment because it already touches. I mean, there will be collision with the protein molecule, I mean amino acids. Okay, so you stop there. So basically, this is the case here. So here for each one, we have a set of potential solution that is generated, and each intermediate level is extended to the next one by adding a new next fragment in all possible conformations. And solution with the bad score are eliminated. Now, if you just evaluate the energy A and B, then you will see that in the case of A, the energy is much lower, that is much negative than B. So then you should choose the option A for that case. So that is one way of doing it. And then there is this stochastic method that you can use. You see, basically here you modify the conformation of the small molecule in the receptor site and assess it on the fly. You can use genetic algorithm. Okay, generally there the torsion angles are added on the chromosomes. Okay, in Monte Carlo based method they are set as parameter for optimization. So we use it quite frequently. But main thing is that generally different orientations that we create by changing this phi psi angle. You see this yellow, these arrows are there. So those are basically the torsion angle of the phi psi angle, that is. So that characterizes the structure of a molecule, okay, protein molecule or ligand molecule. That's how we characterize in chemistry basically. And this has been done by G. Ramachandran. Okay, so he first said that a protein molecule or the small molecule you can characterize using this phi psi angle or the torsion angle. And the whole structural biology is based on this phi. So we characterize each structure by this torsion angle or phi psi angle. It's very unfortunate that he didn't get Nobel, and many people actually recommended it for Nobel, but unfortunately he didn't get it. Anyway, maybe because he was Indian, that's why he didn't get it. And he was a professor in IISc. Okay, so that's how you incorporate it in ligand. Now, how do you incorporate flexibility in protein? Okay, in protein, you do it by fluctuations, that is soft docking method, then side chain flexibility, backbone flexibility, and domain movements. So now, side chain flexibility you can introduce. Backbone flexibility is very difficult to do it in docking, and domain movements is further difficult. But in protein, all these kind of flexibilities are there. So in docking, it's very difficult to treat all these things. So how do you do it? Okay, so one thing is to just use a soft docking method, because here this soft docking method is nothing but the van der Waals energy, I mean the interactions, that is basically soft. It means that it's not stiff, it's flat. The bottom is generally, if you plot it, you see that I didn't show here. Maybe later on I will show you how. So basically the bottom is flat, it's not steep. So that means it can oscillate in that area. Okay, so that kind of thing we do there. Yes, this is the thing. This is the figure basically I was talking about. So here, if you just look at this right hand panel picture, so generally in van der Waals, if you plot it, so this minimized generally is characterized by a single point. Okay, but in soft potential we make it flat. If it's flat, it means there is no energy barrier. No, in both sides there is no wall. It's just flat. So the protein, I mean the atom can move in this plane land because it has the same energy. It can just move around this. So that means when you are inserting your ligand molecule and you see that there is a bump with the receptor protein, then this kind of potential will allow it to move the colliding molecules apart from each other. So there is no collision. Because collision is not possible, because that violates the quantum mechanical thing, no? That's Pauli's exclusion principle. So it cannot be done. So that way you just take care of it. So anyway, so generally what happened, you just do this soft scoring Okay, so you got something like that. Then you do the refinement. The refinement can be done using molecular dynamics simulation, or you can just do the minimization. Okay, just minimize the total energy. You can use, say, conjugate gradient method or steepest descent method, any or Newton-Raphson's method, any method. This optimization method basically you can use to minimize the energy. Then automatically it will adopt a orientation where none will collide with each other. Rather, they can interact with each other via non-covalent interactions or covalent interactions that way. Okay, so as you can see that various linear algebra problem and machine learning things are frequently used in drug discovery. So previously, many of the methods were not available. So that's why it takes very lot of time. Even at that time, computational methods were at the very nascent stage. But last 4 or 5 years, there's a tremendous improvement as method-wise, because a lot of machine learning approaches are now used, AI is used, and not only that, we have now access to large-scale computational facilities. So previously, many of the things we rely on just on raw CPU power, but now is not the case. We parallelize everything in GPU cards, in graphics processing card. Okay, and with this advent of now, I just got email from this NVIDIA. So they have released this 3000 series card, this 3070, 3080, 3090 card. The excellent card, 3090, a graphics card that has more than 10,000 core. Can you imagine one graphics card has more than 10,000 core? You just paralyze there. Your simulation will be super fast and it does not cost you much either. So I have one, before this 3090 came, they had this Titan card. So I had one such card, and then I have actually 2080 Ti card also. So I run everything mostly on graphics card. I use a single CPU. Only one processor I used, and everything is done on GPU actually, and it's much faster. Because 2080 Ti, this NVIDIA graphics card has 4,500. So everything is just mini super. You can imagine. You can use your desktop to run now large-scale molecular dynamic simulations. You can perform it. Now it's a level playing field. Now people cannot say that, oh, I don't have access to large computation, so I can. If you can buy, and this graphics card does not cost too much, maybe 1 lakh or 2 lakhs. Within that, you can buy it. Anyway, so those are different ways that we use it to study these various method, this protein ligand docking. Now, how we do this? The side chain. Okay, so first we talk about this soft van der Waals potential for the flexibility, and now another way is this protein side chain flexibility, because side chain are very is very important, because generally the drug actually they form hydrogen bond or the van der Waals interactions with the side chain. So how the side chain of the protein ligand, the protein amino acids are placed in the binding vicinity is very important. Okay, because that basically tells you, that determines how strong the drug molecule binds. So now how did you do that thing? So studies comparing the X-ray structure of complexes with that of the corresponding unbound protein, okay, the protein bound and bound. So this says that 60% of the side chains modify their conformations upon binding. Okay, so 60% of the side chain, they have a conformational change upon binding. And it has been observed that generally 17 out of the 20 amino acids can be represented by 60. Okay, so now these are
Rangai kita tai यंत्र का जय सिल्वर जो चानी के उपमा में आवेला ओय में कुल जाई.
Can you please mute yourself? Okay. So this approach greatly simplifies the problem and enables side chain flexibility to be tackled. Okay. So you don't need to consider a lot of things. Just you have these 67 side chain rotamers. So you place the side chain into these 67 different rotamer conformations, and then you evaluate the energy and find out for which case the energy is the lowest. So it's very easy then. Okay. So now this is basically then the backbone flexibility. So that is the greatest challenge in molecular talking. Okay. So due to the complexity level introduced by the huge number of degrees of freedom, traditional methods such as the systematic approach or stochastic algorithm cannot be used as a general method. Okay. So this is how you can see that the CPU time it takes the rigid docking to the backbone and side chain flexibility, the fully flexible models. Okay, if your number of parameters increases, this is how the CPU times that increases drastically. So that is the challenge that we are facing. So that is also the part of the problem that we face in computational biophysics problem, the algorithmic problem, because how you can overcome this kind of complex problem. Okay, so we use various statistical methods problem, and also we use machine learning based approach to overcome all these problem. Now one way you can overcome this problem here, this to introduce the backbone flexibility, is to use multiple protein structure approach. Okay, so often if you visit the protein data bank where the crystal structure of the proteins are deposited, you can see that for the same protein, multiple people have solved the crystal structure, and each crystal structure has different resolution. So that means each crystal structure is different to some extent. So that means they are flexible. Nature is you can capture if you consider all this bunch of structure that is already available. Okay, so that's exactly what people are using it. This is called multiple protein structure approach. So for particular one such protein, say CDK2 protein, if we just visit the protein data bank, you will see that it has 7 crystal structure available. So you have the ligand molecule. So you dock your ligand molecules to all these 7 proteins. Okay, so that way you can include the backbone flexibility. But now the question is, if you have a billions of compound and you have 7 structures, can you do this docking for this 7? Because then you are doing it 7 into 1.3 billion thing. So that is also the challenge. Okay, so that is the computational challenges that we face. Okay, so that's why in computer-aided drug design, you cannot. No one can solve the problem alone. We need a concerted approach. We need computer scientists, we need physicists, we need chemists, and we need biologists. So all these 4 people should come together. Then only we can have a successful drug. Okay, so now there are 3 components of docking software. So one is the molecular representation. So that is how you represent the structure and properties, okay, for atomic, surface, grid representations. And then the scoring method. The scoring method is used to calculate the binding energy. Now, if your scoring function is not that good, then the evaluated energy would be bad. So if you just make any decision based on that energy, then of course your decision would be prone to be wrong. So that's why you should have a good scoring function. And the third part is the search algorithm. Okay, you need an efficient search algorithm that decides which poses to generate. Okay, that means exhaustive search, Monte Carlo, genetic algorithm, simulated annealing, taboo search. So all these different kind of algorithm that you can use for searching. Okay, we have to make sure that penalize it. So suppose you generate one structure, you evaluate the energy. Now for the next orientation that you are generating, whether you should, then you see that, okay, for that one, the energy is higher and you don't want that thing. You want to generate a structure for which the energy should go down and down. Okay, so then you are just making it taboo. You are not visiting that one. So that is actually the taboo search. Okay, so that way you can minimize it. Okay, you can make it more efficient searching algorithm that way. So you see that there are a lot of opportunities for a computer scientist to do work in computer-aided drug design actually. Okay. Now, generally, all this docking software is a two-stage docking. Okay, first is the post generation. Post generation means place the ligand in the binding site. Okay, so this is a very well-solved problem. Not much things to be done here. But the research can be done in the post selection. Okay, determine the proper pose. Now you have created a lot of poses. Now which one to choose? That is actually the hardest part, and a lot of this is going on. And how do you determine it? By calculating the binding energy. How do you calculate the binding energy? You calculate using the scoring function. So a lot of research can be done on improving the scoring function that you use to evaluate the protein drug interaction energy. Okay. So post generation, I already told you that it can be rigid, it can be semi-flexible, all different ways that you can generate it. And post selection, where most of the current research are focused, and its more sophisticated scoring functions take longer time. That is the main thing. I told you that in molecular modeling, this is the thing. So you have to decide how much time you want to spend, how much computational resources you have, how much you can invest for a particular problem. Okay, so you always need a balance for speed and accuracy. That is the modeling. Okay, so virtual screening needs to be very, very fast because you are starting from a 1.3 billion compound. Studies on single compound can be slower. Okay, even if you have 10 compounds, then you can use a very strict, very rigorous method to evaluate the energy and rank them further. Okay, then you say, I have used a very strict method and the most efficient method. So I am, instead of 10, I am telling you, you work on these 3 compounds. I am very sanguine that these are the 3 compounds that will act as the best lead molecule. That way you can take that thing. Okay, so that means instead of single stage of docking, we have to use multiple stages. Okay. This multiple stage studies. Okay, so in multiple stages, what do you do here? So, say for example, we are starting with 2 into 10 to the power 6 compound. So we'll do the virtual screening first. We'll use a less accurate scoring function that is called high throughput virtual screening. Glide has a function. So here, if we use 100 CPU, then it will take 2.3 days to screen 2 into 10 to the power 6 compound. Okay, so now using this method, you reduce the number from 2 into 10 to the power 6 to 2 into 10 to the power 5 compound. Now you use standard precision model. So that is better than HTPS. Okay, but now you see that when I say that it is better than HTPS, then you see that in this case, for each compound unit, 120 seconds, that is 2 minutes. Previously, it takes 10 seconds per minute. So 12-fold increase in time. Okay, so you see that. So now it will take 2.7 days on 100 CPU. So using this method, you are reducing the number further down to 2 into 10 to the power 4 compounds. Now you use a method, extra precision method. So that is even more accurate. Okay, even the complex scoring function you are using. Now you see that previously the standard precision takes 2 minutes per compound. Here it is 10 minutes per compound. Okay, so now it takes 1.4 days for 2 into 10 to the power 4 compounds on 100 CPUs. And then you are reducing the number to 2 into 10 to the power 3 compounds. That means 2000 compounds you are downgrading it. Okay, so now you do the visual analysis, further refinement, and reduce the number further and go for synthetic consideration. That is for experimental. Then you hand it over to the medicinal chemist. Now you see that if you use glide XP, like if you use a very complex scoring function or more accurate scoring function in the very beginning, how long it would have take time? Okay, it is 60 times slower than HTTPS. So you can imagine that if you have 2 into 10 to the power 6 compounds and you are using glide XP directly, then you can expect that it will take almost 130 days. Okay, so more than 3 months actually. I mean more than 4 months actually. It's going to take 4.5 months. It's going to take. So, but if you just use this kind of multi-stages approach, then you solve that problem in 6 days. In 1 week, you are done. Okay, so you have to be very judicious how you approach a particular problem. Now, this is for 2 into 6 compound. I am telling you that I did study on 1.3 billion compound. Okay, so there I use recursive neural network based method to do that kind of study. So that is the main challenges and the various applications of computer science in biology. We talk about it. So I'll then so these are so now once you have these 2 into 10 to the power 3 compounds, okay, then what you need to do actually, then you should go for a toxicity testing that you can do using computational based method. Okay, you have a database, lot of database, okay, and you have predicted their toxicological thing. Okay, and now you have this new compound. So how do you do that thing? Okay, you can do it just by it is a simple regression problem. No, you just classify it and then you can predict their these are new compounds and you have the test cases, you already have it. Okay, so you can just use the regression method and you can predict the toxicological properties of these compounds. So these are different ways that we use to study these problems. Okay, so now let us talk about the scoring method. Okay, what are the different uh, the thing is that the scoring method that aims at assessing the quality of docked complexes and guiding the docking algorithm. Okay, so the binding processes that leads to the formation of a complex between a protein and protein means the receptor and ligand is the drug. Okay, is controlled by several factors that first we have to understand. The interaction energies between the two molecules, the dissolution and solvation energies associated with the interacting molecules, and the entropic factors that occur upon binding. The problem is that many of the scoring functions, those are very fast. They don't take into account the effect of entropy. But you know that when the protein and the drug both are free molecules, they both can rotate, they can vibrate, they can translate. But when the drug binds to the protein, then it loses all these degrees of freedom. It loses its translation, vibration, and rotational degrees of freedom. And you know that if it loses its degrees of freedom, that should implicate it. I mean, that should translate it into entropic cost. And that's a penalty because motions are restricted. So that is penalty. So entropy is penalty. Entropy always disfavors the complex formation. Okay. Similarly, the dissolution, the polar solvations. See, when it is not bind with the drug, the protein can interact with the water molecule because in your body you always have water. If it is a cytosolic protein, if it is a membrane protein, then it will interact with membrane. So now, when the drug binds, then it cannot interact with the water. Water are displaced. So that means that will also cause a penalty. Okay, there is dissolution. So you have to. So that is also disfavoring the complex formation. So you have to mind those things. You have to calculate those parts. On the other hand, the van der Waals and the hydrogen bonding or the intermolecular electrostatic interactions, those favor the overall binding energy. Okay, because atom has positive and negative charges, no? So they always will attract or repel each other. So you have to consider all these different interaction part. That will give you the best scoring function. But at the same time, you should remember that if you consider all this possible interaction energy, then your docking will be delayed. Anyway, in your docking, you are not gonna introduce the protein flexibility to the extent it is required. If there is a large scale deformation in your protein due to the protein binding, you cannot incorporate it using any of the docking software. Okay. So then why do you want to use a complex scoring function then? Why don't you use a multiple stages? I use a little bit cheaper method, and then I will cut down the number of conformations that I need to simulate further. So then I'll go for molecular dynamic simulation with these top 3 molecules or top 5 molecules, and then I can study all the protein deformations and whatever going on I can study it. So that will minimize your problem. Actually, you need then less computational resources also, allocations, okay? So you have to have a fine balance when you're designing your scoring function. It should not be too bad that you have identified false negative, okay? False positive. So those which are not the best candidate, but you have identified them, okay? So it should not be like that. On the other hand, it should not be too complex that you need months, several months to screen the database. That is also not advisable. At least in the current situation, this pandemic situation, you must have understood the value of time in the drug discovery process. Okay. So now, generally, I told you that there are these endostatic interactions, hydrogen bond interactions, van der Waals interactions, and hydrophobic forces. And entropy is actually, the hydrophobic forces are actually entropic in nature. So overall, you have to consider these 4.
One can respond to me. I am audible.
Yeah, I can hear you. Can you hear me?
F4, C1, C2. Yeah, okay.
Hello. Should I continue?
Now I am sharing.
We should continue. I think, sir, there is some disturbance in the background.
Okay. Okay. So, so those are the forces that we should take into account. These things, and based on this, the scoring function basically varies from easier to difficult, more complex. So the scoring functions could be shape and chemical complementary scores, empirical scoring, force field scoring, knowledge-based scoring, or it could be consensus scoring. Okay. Shape and chemical complementary score, you divide the accessible protein surface into zones like hydrophobic, hydrogen bond donating, and hydrogen bond accepting zones. And you do the same thing for the ligand surface. Okay. And then find the ligand orientation with best complementary score. Very easy, okay, but it's very heuristic also. It's not the best way to do it. Now the empirical scoring, okay. Here the scoring parameters are fit to reproduce the experimental binding energy, okay. So you have a lot of test cases. You evaluate it, calculate the parameters, and then you assume that these parameters are transferable for the new cases also. Okay, so this is used in Flexes, Ludi, Hammerhead in the softwares. These are used. So here you see that empirical scoring function, they have taken into account all possible things: loss of entropy, also hydrogen bonding, ionic interactions, aromatic interactions, hydrophobic interactions. Everything they have taken into account. Now, the effectiveness of this scoring function depends on the parameters that you have designed, okay, because how good is your test cases, because based on that you have designed your evaluated your parameters and you think that these parameters are transferable for new cases also. That's the thing, but this is very popular actually. And then there is the force field based parameters. Okay, this below one, it has this bond, angle, torsion, and then you have these van der Waals, and then you have the electronic interaction. These 5 different terms. So this is the most accurate method, but this is also at the same time very time consuming, because you can see that you have to calculate the bond energy. So in protein, how many bonds it can form? If it has, say, 2000 residue, or say it has, say, 10,000 atoms. So how many bonds are possible? You have to calculate the each bond energy. You have to calculate the angle energy. Then you have to calculate the torsion energy. Then van der Waals. Then electrostatic interactions. Everything you have to calculate. So it will be very, very time consuming, but it's accurate also. So generally, we don't use this kind of method for this. Okay, so we always need a fine balance between speed and accuracy. For that one, empirical is good enough. And then later on, we will study a supplement our this molecular docking study by large scale molecular dynamic simulation study. So now I told you that one thing, that energy dictates the binding. Now I also mentioned that you have to find the lowest binding energy, and that is your best force. But sometimes it may not be the case. For example, look at these cases. Okay, the lowest energy, I have marked it with this arrow. Okay, that is the lowest energy, and you may think that that is the real binding side, but that's not the case. Why? So what is the barrier height? It is 10 kcal per mole. But you see another side in the middle that has slightly higher energy than the lowest energy. Okay, slightly higher. But the barrier height is 15 kcal per mole. Okay, so the whale is very deep and narrow. So if the ligand or the drug molecule is inserted here, the barrier is so high that it cannot come out. So it will be even more stable conformations than the lowest energy that I have shown you. So in this case, your base structure is this one, the minima, which is separated by highest energetic barrier. That is the base docked pose. On the other hand, you see the third minima. The third minima, you see that it has 8 kcal per mole difference. That's fine, but it is not a good binding site. Why? Because look at how the energy surface looks like. The bottom looks very flat. So flat means the ligand will move around it. It will have multiple conformations, and you don't know for which conformations it will be the best. I mean, it binds most strongly. So that's not a best case. You want that after it binds, it should adopt a single conformations and it binds very strongly. If it moves around in the binding side, that means that's not a good drug candidate. You need to make it a little bit more bulkier so that it snugly fits into the binding site. Okay, so these are the various aspects that you should remember while you want to design a drug. And another thing that you should remember, that which binding site you should target. You have to visualize the protein structure using a visualizing software, and then you have to find what is the best binding site. So there are various deep learning based method also is there. It is called deep talk method. Okay, that is also available that you can use. So you have to find that deep, well-defined pocket, because that is exactly here I show you that this is the best pose for which the pocket is very deep. Okay, shallow pockets have too many options. That is the third case, as I told you, that it is very flat, so it can have multiple conformations. That not good. And then site for specific interactions. Okay, so many charge or hydrogen bond site and mostly hydrophobic van der Waals interactions are bad. So we have to make a fine balance. There should be hydrophobic interactions and there should be charge-charge interactions also. There should be at least one hydrogen bond interactions. That will make even a better drug candidate, okay? And then the well-ordered side chains. Side chains should not be disordered, okay? Because if side chain becomes too flexible, then the drug cannot bind very strongly. Generally, the drug forms a hydrogen bond or hydrophobic interactions with the side chains. So if the side chain is not well ordered, it becomes very flexible, then it's very difficult. Okay. So those are the things that you should remember. And finally, you have to evaluate. Say you short listed your using this method, you have short listed your candidate from 1.3 billion to say 2,000 molecules. Okay. Now from 2,000, you want to reduce it further. You want to move to the 10. Okay. So for that one, then you should use this molecular docking thing, but you have to evaluate the toxicity, and also you have to make sure that this Lipinski's rule of 5 is satisfied. So that is also another filtering criterion that you can use. Okay, what are these filtering criterion? The octanol water partition coefficient is less or equal to 5. Molecular weight is less or equal to 500. That means the ligand, that is the drug molecule, cannot be too big and it should not be too less also. Okay. So it should not be too big. Its molecular weight should be less than 500. And the number of hydrogen bond acceptors should be less or equal to 10, and hydrogen bond donors should be less or equal to 5. Okay. If 2 or more of these rules are violated, the compound might have problems with oral bioavailability. So these are the 5 criterions. You can use it as a further filtering criterion and reduce the number further from 2,000 to less, 10 or 20. You can do it. Okay, so I'll stop here. I'll take a break. Then 11:30, I'll again come back, and then I'll talk about, now you have reduced it to 20 or 10. Now what next? I want to reduce it further. I want to study what happens to the structure of the protein when the drug binds. Okay, what happens to the atomic motions? So we want to study that part. For that one, now we'll go for the second round of talk, that is large-scale molecular dynamic simulations of protein-ligand complexes. So if you have any questions, you can ask me.